For electrophoresis, we use the Beckman-Paragon-Coulter electrophoresis system. For electrophoresis, we need an SPE gel, the B2 barbitol buffer, which must have a pH of 8.6 so that all proteins have a negative charge. The Paragon blue stain is used to visualize the proteins. The protein fixative is an acid alcohol solution, and the destaining solution, which is 5% acetic acid. A normal and an abnormal quality control are also used for comparison with patient test results. The preparation of each one of these reagents is described on pages 2 and 3 of the user's manual. Other equipment needed are the Paragon reagent containers and the gel support. Also required are the gel blotters, the gel templates, and the template blotters, a micropipette, the gel bridge assembly, and the electrophoretic chamber. The chamber is connected to this power supply. The Beckman dryer is used for drying the gels, and the densitometer is used for quantifying protein fractions. Retrieve the patient's specimens as well as the normal and abnormal quality control from the refrigerator. Upon opening the gel, notice that there are 10 application positions, one for each specimen. Review the gel, making sure that there are no dry spots or aberrations on the surface. You are now ready to prepare your own worksheet. The first column has the ID number from 1 to 10. Notice that the sample in well 1 and 10 are the abnormal quality controls, and the sample in well number 5 is the normal quality control. The space between both columns is meant to fix the gel once the electrophoresis is complete, and the right-hand column is for comments on the gel. Before loading samples into the gel, it's necessary to blot the gel to remove the excess moisture. Lightly press on the blotter so as not to create any aberrations on the gel. A sample template may now be applied to the gel. Locate the 2 black dots midway on either side of the gel. Pinch the template and align the 2 farthest slits on the template with the 2 dots on the gel. Lightly press the template down. With the template correctly aligned, sample application can now take place. The sample may be diluted in a 1:3 ratio and applied to the gel, as discussed in the SPE User's Manual, or a smaller quantity of sample may be applied directly to the gel. The latter method is illustrated here. To load the sample, we use a micropipette that has a 2 to 20 microliter delivery. Ensure that the pipette is set to exactly 3 microliters. This is the amount of serum that will be applied to each well. Equip the pipette with the appropriate pipette tip. You'll notice that the pipette has 2 stops, a first stop, and a second stop. The first stop is used for sample pickup, and the second stop is used for sample delivery. Depress the plunger on the micropipette to the first stop. Then insert the pipette tip into the patient's sample without touching the bottom of the tube. Then gently release pressure on the plunger, withdrawing the sample. A small volume of sample can be noted at the base of the pipette tip. The sample can now be applied to the correct position on the gel, depressing the plunger to the second stop. Ideally, the sample should be applied without touching the gel. Capillary action will distribute the sample evenly throughout the well. Follow the same procedure to fill each one of the wells. After all the samples have been loaded, it is important that the gel sit for 3 to 5 minutes to allow proper penetration of the samples into the gel. The electrophoretic chamber must now be filled with barbital buffer. Dispense exactly 45 milliliters on either side of the electrophoretic chamber. After 3 to 5 minutes, carefully blot the template, and then remove and discard both the blotter and the template. Notice that the gel has both a positive and negative symbol. This is used to correctly align the gel in the bridge assembly. Carefully place the gel in the bridge assembly, matching the positive and negative symbols. Ensure that the gel is correctly tucked under the plastic supports. When in place, the gel should appear as a perfect dome. Notice the positive and negative symbols in the electrophoretic cell. Align the gel assembly, again matching the positive and negative symbols. Carefully fit the cover of the electrophoretic cell in place. Insert the electrophoretic chamber into the power source. Once it's in place, turn the power on and ensure that the power source is set at 100 volts. Set the timer for 25 minutes and ensure that the light by the cell number is on. This ensures that there is contact with the power source and electrophoresis will occur. Once electrophoresis is finished, it's time to detect the proteins on the gel. The gel must first be removed from the electrophoretic chamber. It is then placed in the gel frame, which sits in a frame holder. This is what will be used to move the gel between the different solutions. First, the gel is placed in an acid alcohol solution for 3 minutes. This fixes the proteins in place and prevents them from washing out in further processing steps. The next step involves drying the gel in the drying oven. This step is crucial to ensure that your gel is completely dry and there are no apparent wet spots on the gel. Next, proteins are made visible by staining. Place the gel in the staining solution for 3 minutes. In the last step, the gel was overstained by the dye. Therefore, it is now time to destain and dry the gel. This process involves placing the gel in an acetic acid solution for 2 minutes, then an acid alcohol solution for 2 minutes, and then another acetic acid solution for 2 minutes. Once this is complete, the gel must be completely dried in the drying oven. After the gel is completely dry, it is time to interpret the protein bands. This gel shows a typical banding pattern. To quantify these protein fractions, this gel will be scanned by a densitometer. To quantify protein fractions, the Beckman densitometer will be used. The equipment needed is located in this black case. You will find a calibration plate, as well as a blank plate for analysis of our protein electrophoresis gel. Before beginning, it is good practice to ensure that there is sufficient paper in the printer. Visually examine the reading chamber. Ensure that the photo head can move in and out of position. Begin by turning on the power switch, located at the back of the machine. The first screen that appears requires the entry of today's date. The main menu displays the different types of electrophoresis scans. Before scanning any gels, the machine must be calibrated. Select A for SPE gel, and then select C for calibration check. When the message appears, select Yes to proceed. Insert the calibration plate, extend the photometric head, and close the cover to the reading chamber. Calibration will now take place. In a few minutes, the results of a fluorescence and absorbance calibration are printed. Once calibration is complete, remove the calibration plate. Align the gel on the blank plate using the magnets to hold the gel in place. Carefully fit the blank plate in place. Extend the photometric head and close the lid to the reading chamber. From the main menu, select A for protein electrophoresis and A again for all scan. When the work list screen appears, enter the patient identification number and the amount of total protein in grams per liter. Enter this information for each patient sample. When the work list is complete, push the Start Scan button on the keyboard. When the screen indicates that the scan is complete, press C to review the results. To print the results, select P, and then select the number of the gel to be printed. The results for each patient sample will be printed. The bands on the gel can be divided into 5 major fractions: albumin, alpha-1, alpha-2, beta, and gamma. The relative density of each protein fraction on the gel is translated to a graph such as this one. An increased density of a protein fraction will give a higher peak on the graph. This shows the results of a normal protein electrophoresis. The results obtained should be compared to the reference ranges to detect any abnormalities, especially the gamma protein fraction. The results seen here are for the protein band posted above the screen. The first column on the left lists each protein fraction. The second column lists the relative amount of protein in each one of these fractions. The final column shows the absolute value of protein in each fraction based on a total protein of 85 grams per liter. In this example, it is to be noted that the gamma fraction has a much higher peak than is normally observed. This increase indicates a paraprotein, or a monoclonal gammopathy. Upon further examination of the absolute and relative values of the gamma fraction, we see that it is very much elevated, confirming a paraprotein. An immunofixation technique may be done on this patient to identify the type of monoclonal antibody present. For immunofixation, the gel preparation and electrophoresis are similar to the steps outlined for serum protein electrophoresis. When electrophoresis is complete, the gel is removed from the electrophoretic chamber and anti-sera must be applied to the gel. First, align the IFE anti-sera template with the 4 corner position dots. Gently press down on the template to ensure its position. Next, apply 80 microliters of anti-sera to the appropriate well. Place the gel and template into an incubation box and incubate at 45 degrees for 30 minutes. When the incubation period is complete, discard the anti-sera template. Place the gel in a gel frame and place the whole assembly in a saline solution for 10 minutes. After 10 minutes have passed, remove the gel from the saline solution. Prepare the gel press for drying by placing one gel blotter at the very bottom of the press. Lay the gel on top of this blotter and cover the gel with another gel blotter. Place 2 drying blotters on top. Replace the cover by aligning the pins and place the weight on top. Press the gel dry for 3 minutes. Upon removal of the gel from the drying assembly, there should be transfer of the excess anti-sera from the gel to the blotters. Remove the gel and soak it in a second saline solution for 10 minutes. After the second wash, blot dry in the same fashion as before for another 3 minutes. Remove the gel from the gel drying assembly and place it into the dryer until completely dry. This should take approximately 5 minutes. After the gel is completely dry, place the gel into the same holder assembly that was used for protein electrophoresis. Place the gel assembly into the Paragon blue stain for 3 minutes. After 3 minutes in the stain, remove the gel and immediately transfer to the 5% acetic acid solution for 2 minutes. Transfer the gel to another 5% acetic acid solution for another 2 minutes. This step will clear all the remaining background stain from the gel. Remove the gel from the acetic acid solution. Remove the holder and note the clearness of the gel, as well as the precipitant bands. Place the gel into the Paragon dryer until completely dry. This may take 10 to 20 minutes. Once complete, the gel will show a banding pattern, such as the one illustrated here. Note that the albumin has migrated towards the anode, which has a positive charge, and the immunoglobulins towards the cathode, which has a negative charge. The SPE row should match the electrophoresis pattern obtained on the SPE gel. This patient has a sharp paraprotein band in the gamma region. To identify the type of chain and immunoglobulin, the paraprotein should align horizontally with at least 2 of the other columns, which will show a band in the same region. In this case, the pair protein in the SPE column aligns with a dark band in the IgM column and a dark band in the kappa column. Therefore, this protein can be identified as being an IgM type kappa.